Okay, in this video, we're going to talk about the processing and the function of microRNAs, or miRNAs. And what these microRNAs do is they regulate gene expression via regulatory RNAs. Now, this process is sometimes referred to as RNA interference, especially when we use it in the lab as a way to knock down particular messenger RNAs. It was discovered in the 1990s. It won the people who discovered it the Nobel Prize. And there's lots and lots, there's about 474 microRNA genes that have been found in humans. And it's thought that they regulate about 1/3 of all human genes. And these microRNA sequences have been conserved across distant species. So the genes that encode these microRNAs have been around for a while. So in the most basic level, here's what they do. So after a microRNA is processed, it becomes part of this protein complex here. So here's another ribonucleoprotein complex. And the microRNA itself is reverse complementary to specific messenger RNAs in red here. Now, if they are perfectly reverse complementary to that messenger RNA, then what happens is those messenger RNAs are degraded and therefore can no longer give rise to protein. If the microRNA has an imperfect match to a messenger RNA, it still binds, but instead of catalyzing the destruction of the messenger RNA, it simply inhibits translation from occurring. One way or the other, what microRNAs do is they stop messenger RNAs from being able to be translated into protein. So they stop protein production. And in this way, they are regulating gene expression. Well, microRNAs, like any other RNA in the cell, is encoded by a gene within the DNA. So here we have RNA polymerase II, and it transcribes a gene that encodes a microRNA. Now that microRNA is just a very small part of the RNA that gets transcribed, and so it needs to be processed. And the first processing event is this stem loop that microRNA is part of here, that self-binding part, is cut from the RNA by an enzyme called DROSHA. And so we go from our primary microRNA to our precursor, our preliminary microRNA. Now that its precursor microRNA gets exported into the cytoplasm, where now it is the substrate for an enzyme called dicer. And dicer nips off the stem loop portion here, leaving us with double-stranded RNA. So this is an unusual molecule, right? This is actually two strands, separate strands of RNA that are reverse complementary and binding to one another. We call this short interfering RNA, or duplex microRNA. Now, one of the two strands is the actual proper microRNA. That's called the guide strand. It's going to be reverse complementary to the target sequence, to the messenger RNA. But the passenger strand needs to eventually be degraded. So that's the job here of the RISC complex and AGO2. So this double-stranded, this microRNA duplex is recognized, the passenger strand is degraded, and the guide strand is incorporated into this RNA-induced silencer complex, or RISC. And so now we say that RISC has been programmed. It's bound to this microRNA, and wherever that microRNA is reverse complementary to, it will bind and inhibit the translation of that target messenger RNA. Again, if it's an imperfect match, it'll just block translation. If it's a perfect match, it'll catalyze the degradation of that messenger RNA. Now, RNA interference, has been a crucial tool for research. And what we can do is, because all cells have DROSHA, all cells have dicer, all cells have this RISC protein, that if we incorporate RNAs of our own, microRNAs of our own design, we can specifically target messenger RNAs to stop the production of whatever protein we want to stop. Those are called knockdown experiments. We're knocking down the production of a specific protein. Now, one of the best ways to do this is to transfect cells with an expression vector that expresses a primary microRNA that will then be processed into a microRNA, a mature microRNA that will then knock down whatever protein we want to knock down. Now, the paper that you guys are reading really takes advantage of this RNA interference to knock down the production of UTX, one of the proteins that they're studying. Let's take a look at what question they're trying to answer by using RNA interference. What they have already shown in the paper is that retinoic acid leads to the activation of HOXB1. They have also shown via chip experiments that retinoic acid leads to the accumulation of trithorax here at this particular region of DNA just upstream of HOXB1, and specifically not trithorax, but more specifically JMJD3 and UTX. So they showed two things. Retinoic acid is necessary to turn on HOXB1, and retinoic acid is required for attracting UTX to this region of HOXB1's promoter. What they have not said is this. What they haven't figured out is retinoic acid independent in its attraction for trithorax to the promoter and turning on HOXB1? Are those two independent activities, or does it work this way? Does retinoic acid attract trithorax and UTX, which then demethylates K27, which then attracts the activators? That's the hypothesis they want to propose. They want to propose that the activation of HOXB1 expression by retinoic acid is through the activity of UTX, which means that if you get rid of the activity of UTX, even in the presence of retinoic acid, we're no longer going to get HOXB1 turned on. That's what they want to say. So that means they need a way to turn off the expression of UTX protein. They want to get rid of UTX protein and show that even in the presence of retinoic acid, if you remove UTX, you no longer get the expression of HOXB1. So let's take a look at a few experiments that they do. So here we're looking at, I believe this is 3C. And here we have SHUTX1, SHUTX2, short hairpin UTX1, short hairpin UTX2. These are expression vectors that have a gene that encodes a microRNA that will knock down UTX expression. That's what this SHUTX1, SHUTX2. They've made two different constructs. They weren't sure which one was going to work. And then this is a short hairpin. This is an expression vector that expresses a short hairpin scramble. This is our negative control. Okay, so they have 3 cells, and let's take a look. They are NT2D1 cells. These are eukaryotic mammalian cell line. One of the cell lines expresses this microRNA. One of them expresses this microRNA. One of them expresses this microRNA. And now what they're doing is they're taking these cells, they're either, okay, so let's take a look at this cell, the cells that express our negative control, the RNA, microRNA that targets scrambled messenger RNA. One batch of these cells, they incubate in the presence of retinoic acid. One batch of the cells, they incubate in the absence of retinoic acid. They then lyse the cells open, and they use quantitative, reverse transcriptase, quantitative PCR to measure the amount of UTX messenger RNA within those cells. And what you notice is that really whether or not there is or is not retinoic acid around, the amount of UTX messenger RNA is pretty much constant. And pretty unaffected by retinoic acid. Okay, so retinoic acid doesn't seem to play a role in how much UTX messenger RNA is going to be made and how much protein is going to be made. What retinoic acid appears to be doing is it's localizing that UTX protein to the promoter of HOXB1. But that's in a different experiment. Now this is our control. This is our negative control. It's just basically showing us how much UTX messenger RNA there normally is within a cell, whether we have the presence of retinoic acid or the absence. And now we look in the cells that are expressing microRNAs that are intended to knock down that UTX messenger RNA. Does it work? Heck yeah, it does. Look at this. The amount of UTX messenger RNA, whether we have retinoic acid, whether we don't have retinoic acid, is extremely reduced, regardless of which construct we're using. This is simply a control. This is simply the authors demonstrating to us, the readers, that their microRNAs, their short hairpin RNAs that are intended to knock down UTX messenger RNA does indeed knock it down. Here is, in the absence of those microRNAs, here's the expression of UTX in the presence. So, it's not 100%, but it's pretty darn effective. Now, what we want to do is remember, we want to see, is UTX, which is part of trithorax here, is UTX presence necessary to turn on HOXB1 expression? Right? Basically, what they're asking is retinoic acid going through UTX to turn on HOXB1, or is retinoic acid independently turning on HOXB1 and localizing UTX? All right. If it's this first one, where it requires UTX, then we should be able to knock down UTX, and even in the presence of retinoic acid, HOXB1 no longer is expressed. Let's take a look at that. So now we are looking at the amount of HOXB1 expression in cells that have either been incubated in retinoic acid or not incubated in retinoic acid. And here's our negative control again. These cells are these NT2D1 cells that are expressing our microRNA that doesn't target anything related. And we get what we expect to get. In the presence of retinoic acid, HOXB1 is turned on. Beautiful, we've seen this already. In the absence of retinoic acid, HOXB1 is not turned on. Fantastic, this is great. Now take a look at our experimentals. Okay, so without retinoic acid, yes, we should expect no HOXB1 messenger RNA, yes. But now, when UTX messenger RNA is being knocked down, we also get a lessening of that HOXB1 activation by retinoic acid. Now it's not full, but it's pretty darn effective. And the fact that it's not full, that it doesn't, that's getting rid of UTX doesn't completely stop HOXB1 expression, probably has something to do with the idea that knockdown experiments, when you're knocking down messenger RNA using RNA interference, isn't 100%. A better experiment, but it's more costly and more time consuming, is to delete the UTX gene in cells so no UTX is being made at all. It's expensive, it's time consuming, this is much easier to do. So what this is showing us, and what this seems to be demonstrating, is that retinoic acid's activating of HOXB1 and turning on HOXB1 relies on the action of UTX. Without UTX, retinoic acid doesn't induce the expression of HOXB1. And that's what these experiments seem to demonstrate. Now, the question is, does or is UTX necessary for the demethylation of K27? So let's take a look. This is the most complex figure in the entire paper. First of all, let's just make sure we know what we're looking at. This is a chip experiment. So we're back to looking at how much protein, which proteins? HA, H3K27ME3, and just any histone H3. What proteins are associated with the DNA upstream of HOXB1? So DNA at the promoter region of HOXB1. So we're looking at a chip expression or a chip experiment, and we want to know how much protein is bound to this particular region of DNA under given circumstances. And so we have a whole bunch of controls here. We're looking at cells that either express our negative control microRNA that targets the scrambled messenger RNA, or our experimental cells that express a microRNA that targets UTX messenger RNA. In those cell types, we are first doing our control where we're doing a chip experiment with an antibody that binds to the hemagglutinin protein. Now, hemagglutinin is not expressed in these cells, and therefore we don't enrich for any DNA, any of the HOXB1 DNA. Good, that's our negative control. Here's another control. We do that same experiment, and we see that regardless of whether we are knocking down UTX or not knocking down UTX, we have the same amount of histone H3 bound to the promoter of HOXB1. The same amount of histone H3. So this is like we know that we're not changing the amount of histone H3 sitting at this promoter. So what are we changing? Well, look at this. In the presence of the UTX microRNA, so that's here, right here, we have the UTX microRNA, which means we don't have any UTX protein. Look how much of our histone H3 is trimethylated. Quite a bit. But then when you add, when you allow the UTX protein to be made by not having the microRNA there, by not having the UTX microRNA that targets UTX, which means we have lots of UTX protein there, now we have a lot of demethylation. We have less trimethylated, which means this supports the idea that the demethylation that we see of K27, of histone H3, is indeed coming from UTX. So what these experiments are showing us is that what seems to be happening is this. Retinoic acid comes in, that attracts the trithorax complex, including UTX. Then UTX demethylates K27, which attracts the activators. We don't know what those activators are yet, which does something, which then turns on HOXB1. That's what these experiments are showing. Now, they do the same sorts of or similar experiments in using immunohistochemistry. So in experiment 1C, what they show us is that they take HeLa cells co-transfected with short hairpin RNA to UTX, so that's their shUTX1, and a PCMV H2B GFP, so basically another transfection vector, expression vector that makes GFP, the green fluorescent protein. And this is just a way to determine which cells were successfully transfected. So in this part here, what we're looking at are here, DAPI stain, meaning it shows all of our cells. GFP means these are the cells that are expressing our short hairpin UTX1. And now, if we stain using an antibody that binds to UTX protein, look at this. The cells that express our microRNA, boom, 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 do not make UTX protein. Empty, 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 empty. Brilliant. That's a control that works the way they want it to. Now, the question is, if we don't have UTX protein in these cells, is there going to be more trimethylation because there's no demethylase? Let's take a look. So here are all of the cells. The GFP shows us the 1, 2, 3, 4 cells that are making our microRNA and therefore lacking the UTX protein. So let's take a look. Compare 1, 2, 3, 4 cells and their ability to produce or their amount of the trimethylated K27, and sure enough, yep, there's more trimethylated K27 in these cells than there is in the cells that make UTX protein. So here is independent verification that yes, UTX protein is necessary for the demethylation of K27.